And hello, good afternoon, good evening, wherever you are in the world. We're having a LinkedIn and Facebook Live interview where I have the incredible pleasure of speaking with Vanya Loroch from Switzerland, a molecular biologist. And we have him here, and he's going to be sharing some of the breakthrough concepts that he has done with inhaled Losartan. So the first thing we want to do is that I want to give a bit of an introduction, let him introduce himself, and then I'll be saying a disclaimer afterwards in relation to his work. So go ahead.
Yes, thank you very much, Philip, for the opportunity to share what we have to share. I believe it's going to be interesting, exciting, intriguing, definitely out of the box. And I'm a little bit of an out-of-the-box guy. I'm a molecular biologist. For the last 15 years, I have been helping decision makers, a lot of them in big pharma, but not only, essentially business people, non-biologists, actually all non-biologists, to get a better understanding of the biological foundations of what makes their companies, what makes their institutions happen. So I am a teacher at heart, and I certainly didn't expect to endorse a new role today, which is that of trying to bring a real practical solution to a hell of a problem that we're all facing.
So let me clarify. So my involvement in this is quite random, in that I specifically mentioned Dr. Annette Offringer. Now, I consider her a critical expert in terms of what she's been doing from the Netherlands, and I hope to be able to speak to her again soon about her data. But when she was talking about the receptors, the ATR1, ATR2, which I think you'll talk about some more, I had just mentioned in a post on LinkedIn that wouldn't it be good to have an inhaled angiotensin receptor blocker? And remarkably, you responded, I will get onto it. And within 48 hours, you had a solution, which blew my mind. And I thought, wow. So I have no connection with anything you're doing, except for the fact that I realized that I was in contact with somebody who can make things happen very quickly. So how in the world did you do that from just an idea to a product within about 9 days?
Yeah, actually, yeah. So thank you for Philip. Actually, my merit, I don't have very much merit to it, but I do happen to be part of a project which originated with an invention that took place in Novosibirsk in Russia, and my partner. We are a small, call it a startup company, and I'm not a commercial guy, I'm a scientist, but I got involved in this project because I liked the science, and the inventor of this technology, actually he's the one that deserves to have the credit for all of this, and we were basically, at the beginning it's a material science company, so the idea was to take chitosan, which is a very, very well-known substance used in all kinds of industries, and to try to make something better out of it, of making nanoparticles, and if we have the time, I can explain a little more into it. So I became interested. I helped. I'm based in Switzerland. Russia's far away. I happen to be of Russian origin, so I made a connection. I made a connection to Switzerland, and then when Annette, started discussing with me her vision of what may be going on with SARS-CoV-2 and the ACE2 receptor and the AT1R, I had to do some very quick learning, to be very candid. I think I'm not the only one. Really, all of us have to do very quick learning. So I learned that bit, and then I reached out to my colleague, his name is Vladislav Fomenko, and I said, hey, there is this idea that in fact angiotensin type 2 blockers Losartan, Valsartan, others, this may be a good thing to actually try out and see whether that can help alleviate and counter some of these dramatic inflammatory responses we see in some patients. And he just jumped on the wagon and made the aerosol was ready the next day. So he blew my mind away, and he said, "We're ready, let's try it." And I basically said, that was a bit fast. I'm not ready. I don't know what to make out of it. So this is actually how it all started, and it's been a very, very fast-paced learning experience, sharing, meeting you, Philip, discussing this with Annette, and I would like to say the rest is history, but the history is in the making.
History in the making. So let me just try and see if I can give a little bit of explanation, and I then want you to clarify it a bit. So, as far as we know, there is a renin-angiotensin system in the body that functions to manage blood pressure to some degree, as well as inflammation in the body. And it's a balanced system. And you have, critically, in the context of the COVID crisis, there are two receptors that are relevant: the ATR1 and the ATR2. Now, it seems as though both receptors work together, but what's unusual is that the COVID, the virus, the coronavirus here, specifically binds to the ATR2 receptor, and then it gets into the cell. And it's through this process of potentially imbalancing the system that we're having lots of complications. Would that be about right? What do you think?
Yeah, well, my understanding, first of all, I think the exact mechanism is still not clear. I mean, we're trying to put these pieces of the puzzle together. I think with those receptors, there's also one of the central pieces, which is the ACE2 enzyme itself. So my vision as a molecular biologist is that these different membrane proteins, call them receptors, as far as the ACE2 is a receptor for the virus. So I guess all of this business comes together, and depending on the ratio of AT1R, AT1R or AT2R, you get endocytosis with the virus that actually, hitchhikes this whole system, gets endocytosed, replicates inside the cell, and as a net result of this infection, ACE2, which converts the pro-inflammatory angiotensin 2 into the anti-inflammatory angiotensin 1,7, that particular enzyme disappears from the surface of the cell, and we get these exacerbated inflammatory response. So it's a loss of homeostasis of these different players. Now, so the exact sequence is not clear, I think, or at least I don't know. Maybe Annette, clear it out with Annette next time.
Yes, we'll get Annette again soon. But in effect, so two receptors. This one here is the one potentially that the COVID virus would bind to. This one here is the one that the Valsartan would bind to. It seems as though both work together. So when the virus is on this one and takes it away, all you've got left is this one, which is pro-inflammatory, and then causes a lot of the problems, I guess, in the lungs and in different parts of the body. And so Losartan, what does that do? Does that then block it?
Okay, Losartan will bind with a very, very high affinity to the AT1R receptor.
So not the one that the virus binds to, the other one.
Right. But by doing that, it will actually inhibit the endocytosis of ACE2.
Yeah.
So in the presence and by doing that, it also upregulates the expression of ACE2. So there was a bit of a controversy in the beginning because some people claim that Losartan and other ARBs will actually jack up ACE2 expression. You'll have more receptors for the virus, therefore the virus will become more virulent. So was the argument. Don't take Losartan. All these patients that have hypertension, they should get off Losartan because it could be dangerous. It would make them more susceptible to the viral infection and getting out of control. However, by inhibiting AT1R, you keep ACE2 on the surface, and that is anti-inflammatory. I guess the take-home message, it's not the virus that is actually putting directly the people in the dire condition of having the lung tissue destroyed. It's the inflammatory response. So it's the excessive inflammation. Therefore, we now know there is mounting evidence, I think daily, that the more ACE2 is present, the less the risk of having respiratory distress. And the less AT1R you have on the cell surface, the more ACE2 you will have. So Losartan and other drugs are actually protective.
We'll just continue talking about this, but this is a very important piece of the puzzle. So in effect, we've just reached the point where you had some degree of a block. So the Losartan will block the ATR1 receptor, and this will therefore inhibit the inflammation.
It will inhibit the inflammation, and at the same time, we believe, although that remains theoretical at this point, that will interfere with the endocytosis via ACE2 of the virus, so it will actually act as an antiviral indirectly. Wow. So it's a double hit. So the idea, it's a double hit. It will prevent viral, it will interfere with viral entry into the cell via endocytosis, and at the same time, by preserving ACE2 on the surface, it will combat the excessive inflammation we see in patients with respiratory distress.
Yeah, so let's see. So we have two levels that the Losartan can act to block the viral entry into the cells and protect the body from the inflammation that occurs within that renin-angiotensin system. Correct. And so therefore this, in theory, are there any risks associated with what you've done?
Well, I understand that one of the issues that patients that are in experiencing respiratory distress and are in an advanced stage of this disease is that they may already have low blood pressure. Yeah. And that's one problem. And the second problem is that if they are on a respirator, on a ventilator, they cannot take drugs the oral route anyway. Yes. So if you want to bring Losartan, or Valsartan, or in fact any other drug that you want to work in the lungs, you don't necessarily want to go to the systemic route because you're going to have you're going to have side effects, you're going to have systemic effects, which may actually make it very difficult for dosage or for whatever reason. So the idea is actually pretty simple. Can we deliver a potent drug to the location in the organism what it creates, where the virus is creating the most damage? And that is obviously the lungs, the lower respiratory pathways, and then, of course, upstream, the upper respiratory pathways. Yeah. My understanding, and I may be wrong, but this is where, and I think we all agree that this is where the acute problem is. There are others, because the virus does damage elsewhere in the body, but this is the major emergency problem that we need to deal with. Therefore, the idea of delivering drugs via aerosols, and this is where our technology perhaps can help.
And so when you say an aerosol, Do you mean a typical inhaler? Is it some special machine?
Okay, so the nanoparticles that the aerosols that we have created, they are generated by a ultrasonic nebulizer.
Yeah.
So you can have a portable device. I don't know that much about the technology, but the idea, at least for the critically ill patients, is to connect that aerosol to a ventilator, to a respirator as an inlet, so that it actually goes inside the lungs and is delivered over a period of time.
Yes. And so in a well person, do you imagine that it could also be delivered? When I say a well person, so if you imagine someone who has the early stages of COVID and you still want to inhibit viral replication inside the upper airways, do you imagine that it could be delivered by either nebulizer or even an inhaler?
Yes, I believe so. I mean, this is more of a technical question, but I don't see why not.
Yeah.
I don't, just like any other inhalable drug, like, in asthma or any other disease. In fact, the reason why I think where things get a bit tricky is if you want to actually cover the entire 100 square meters or so of the epithelial alveolar tissue in the lungs. Now, if you want to cover all of this, then you need to make sure that your aerosol droplets are small enough, but not too small, so that they actually stick and cover the entire surface. But if it's just about trying to get it in the upper airways, I assume that it is actually less tricky because the surface is smaller, it's closer to the application, etc., etc. But then it becomes almost like some physics of small particles.
So one of the things, so where are you in the context of this work? So from day zero, which is probably 9 or 10 days ago, when you had the idea to do this, till now where you've got a product, what is the name of this product and what are you planning to do with it?
Okay, if I can share my screen, I will go ahead and run you really rapidly through this. Tell me when you actually see the screen.
Yeah.
Okay, so there it is. Okay, it's working. It's better, much better than last time. So the name, so the name of the product, and I'll just it's Novocaizol. It is a trademark, but it's basically chitosan. Now, chitosan was actually discovered or invented by Charles Rouget. We're talking 170 years ago, 161 years ago.
You're breaking up a little bit again, Vanya.
Okay,
Let's make sure we've caught that.
So chitosan is a very old molecule used in all kinds of applications. The problem with chitosan, it's a linear molecule, and it's not soluble unless the pH is acid. So it has limitations. So what we have developed is a technology which takes this linear chitosan, goes through 2 steps where it basically activates the side chains of this polysaccharide and activates this, and then we can add an active ingredient, which can be a small molecule or a protein. It's pretty neutral, and then we actually engage this construct into an intramolecular reaction, and we create a little nanoparticle which basically acts as a sponge. Okay, and that's the technology. It works. It's simple. The nice thing about it is just chitosan. There's nothing else. And chitosan is a food additive. It's recognized as grass. So it is something which is which we believe is very safe. That being said, we don't have the clinics done. So that's that. So it has all kinds of applications. But one of them, and this is what we're talking about, is really drug formulation, drug delivery. So here, this is how I envision Losartan formulated with these nanoparticles. Okay, so the key feature here is that, first of all, it makes the drug soluble. In other words, it's a nanosuspension. So we solve the problem of being soluble or not. It's a suspension. The second feature, and that is key, it's bioadhesion. In other words, these particles, once they land on a tissue, mucous membranes, inside muscular tissue, and also in the lungs, they stick. They stick and they stay. So this enables localized drug delivery. And by the same token, sustained release, with no systemic distribution of the active ingredient, or very, very little, because the dosage, there's no comparison with oral drugs, because it's almost two-dimensional as opposed to filling up the body. And because it's protected for enzymatic degradation, we believe that it is a very nice way to deliver a drug locally, even at high concentration.
And this will go all the way down into the lungs.
Correct. So because these the droplets we create in the nebulizer, they're actually dry material because it evaporates. The size is 200. Average size is about 300 nanometers. And it is just the right size so that it goes everywhere, and it's not too small so that it actually lands on the tissue and sticks. So we believe, and there's anti-inflammatory properties which are inherent for chitosan. They're well known, and we can enable these multiple formulations. So we have this technology, and we decided to go with Losartan, thanks to this learning experience that I had with Annette, and that I think we both shared, and I believe I'm willing to bet all the money I don't have on the fact that she is damn right.
Yes.
So I really believe I don't know if it's intuition, because she is, her background is general practitioner, interested in disease mechanisms. Well, talk of thinking about out of the box and coming up with something. So yes, so I'll skip this part because we already discussed this. But the bottom line here is that we decided to formulate Losartan, Valsartan. So we have that. We have a stock of that. We have Valsartan, and what we did immediately, we said, okay, let's simulate it. Let's send it into a test animal, which is a swine, and understand, miniature pigs are routinely used for models for human lungs. So the swine, I nicknamed it Gagarin, the first man in space, right? Because it happens to be Russian, and it's a first. And we actually simulated as well as we could a situation of a patient who is intubated. So we intubated the pig, anesthetized the pig, connected the pig to a ventilator, and loaded his lungs with these aerosols. And we basically loaded probably something like 10.
So we had the swine. So you actually put the aerosol in the lungs.
Yes. And as we did that, we actually delivered 10 to
a little bit. This is why. So sometimes you're
Yeah, I don't know what's going on, because I usually have a good internet connection, but maybe it's I don't know. Can you hear me okay now? I guess you can't. I guess you can't. I'm very sorry for that. I don't know what to say.
So in effect, so you gave the pig and the anesthetized pig into the lungs.
Yes.
Yes.
Yes, and and then we monitored, of course, what we could monitor in real time, meaning blood pressure, key factor, and also hemodynamics, and everything is absolutely normal with the swine. In other words, we were able to demonstrate that locally the drug was there, systemically it was absent, which is exactly what we wanted to see.
So you were saying that once you've got these inhaled particles, you then anesthetize the pig, put it in a ventilator, and put it into the lungs of the pig. Yeah?
Correct, absolutely.
Were there any problems with the pig during that?
None whatsoever. So we measured the basic vital parameters of the pig. Blood pressure,
yes,
normal, didn't change. The same before, the same during, the same afterwards for a week.
Yes.
So the drug did not go systemic.
Right.
The other thing we measured is hemodynamics. Nothing. So that was a good sign. And we are now carrying out experiments on rats that will be sacrificed, euthanized, in order to see actually in situ, do some histochemical microscopy analysis to try and visualize where the nanoparticles, how they cover the lungs using fluorescent markers. So we are doing all this preclinical work right now.
And so you're actually going to do a study soon with this?
Yes. So then what happened, and this is where things become challenging and important and so critical. We have initiated a discussion with some clinicians who are right now unfortunately desperate because some of their patients are in fact dying. And the argument of Losartan as being a decoupler of inflammation and viral infection, they deemed it to be convincing enough. And they believe, from what I shared about chitosan nanoparticles and our experiment with the pig, that actually this is worth a try, and we hope to launch sort of a last chance small-scale clinical trial for these critically ill patients in a major research hospital in Europe. And we are looking for the clearance from obviously the hospital. This is a compassionate use last chance scenario. And, we're just going to have to go for that because unfortunately the stakes are so high that there is probably not much to lose. And, it's a complicated decision. It belongs to the clinicians. It doesn't belong to us. All we can do is support them.
Yes.
So absolutely. We should know. If we get to do this clinical trial, we should hopefully see a difference. And the dramatic part, and I think it's important to share that. Naively, I ask, what are the endpoints of this clinical trial? The answer, unfortunately, was, it's very simple. It's death or life, so it's binary. And we do what we can with what we have.
I understand. And so let's just think practically now then. So if you've created this product, just imagine that this project is beneficial in early and even later stages of the disease. What is the reality for countries, because suddenly it would become extremely expensive. Is there any way to make sure that that's accessible to as many people as possible?
Well, that's an excellent question. There's perhaps a business side that I'm not going to get into because that's not me. But all I can say is that the raw material, chitosan, is widely available. It's dirt cheap. Obviously, there are different grades. The chemical synthesis is very, very simple. It can be done in a matter of hours. It's scalable. So it's a simple chemical reaction. It's not complicated. It's a bit smart. It's a trick. I mean, chemistry is a bag of tricks, with a theoretical foundation. So the ingredients are affordable. They're inexpensive. They're widely available. If we look at sartans, these are easy drugs. Clearly, we are not in the same game as say big pharma developing very sophisticated, innovative molecules which are very expensive and protected by IP. We are in a different space. So I think, I would like to see it given a chance. I'll stop there. There. And there are others. Losartan is only one of potential candidates. But there are other drugs that are probably worth a try, and systemically they may create challenges, but locally these challenges may be overcome. And I'm thinking in particular about toxicity and undesirable off-target effects that we don't want. So there are others.
No, we've been going quite a while simply because of the breakups, and this is absolutely fascinating. But one of the things I wanted to capture before we finished was to do with some of the work that you've done with digoxin. What is that about?
Okay, well, digoxin is one of those cheap, old, well-known drugs used, in cardiology, in heart attacks and arrhythmia and other conditions. And interestingly, digoxin popped up in two very different screening protocols. The first one is a paper that was just published by a team of South Korean researchers, where they actually looked and screened 5,406 approved compounds in a in vitro cell assay using MERS-CoV virus as a model. And digoxin popped up, not the only one, but one of the few candidates where they saw biological activity, where basically they saw that the MERS-CoV was inhibited from being expressed inside the cells. So that was a cell assay, and this is something which is not that new. It was on the radar when the MERS epidemic happened. Also, I understand something with Ebola. So there are these compounds that have antiviral activities.
Right.
And the second study, completely unrelated, was actually in silico virtual screening. And it's an Indian team that looked at a huge number of compounds based simply on bioinformatics. And did I lose you?
No, I'm right.
Okay.
Here.
So the second work was completely different, based on the docking, on measuring the docking forces between the receptor And in fact, the receptor here, or at least the molecule that is being targeted, is the SARS-CoV protease. It's the same enzyme that is present in HIV, in other coronaviruses, that actually cleaves the polyprotein, which is produced by the virus, into the components that make up the viral capsule.
So in effect, it's another one of those products that can inhibit the replication of the virus inside the cell.
Yes, it's a true antiviral, as opposed to Losartan, which works on the human component of the story.
Yeah.
So it is a toxic molecule, so the therapeutic range is pretty narrow when taken orally. We believe, however, because it's non-systemic, that we can probably crank it up.
Yeah. So meaning that you would put it again in an aerosol and put it in the lungs.
Yes, it's done. We have the aerosol. We're doing some toxicology studies on rats, because unlike Losartan, which is pretty safe, and we know that digoxin is a more tricky one, because it can be toxic.
A more difficult drug.
Yes.
Listen, Vanya, this has been an absolutely fascinating conversation. I am so sorry we had those interruptions, because I think that the information that you've shared here, one, gives us hope that we can find solutions really quickly when you have the right people using the right kinds of technology. And two, what really inspires me is that this stuff you're using is readily available and cheap to make, which is one of the biggest concerns, is that in second and third world countries, if you come up with something that is expensive, they can't afford it, and
Absolutely.
they have probably a higher need for some of this stuff. So I would like to hope that this work continues, and I would love for you to keep us updated on the outcomes of some of those clinical trials.
Absolutely. We are sharing this, we are open, there's obviously, this is not commercial at this point. We really want to bring this technology and make it available. And obviously alone we cannot do that. So we need clinicians.
Yes.
We're looking for organizations, whether they are pharma, whether they're research hospitals, it's secondary. We're looking for organizations that can help us do it right in the context of what we're going through.
Yes.
Thank you very much, Philip, for the opportunity. There is a website, and I don't know if you can post it, but that's kind of the interesting part. There's this website I created on the spur of the moment, which is a repository. It's of everything that is happening.
What is the www for it?
So it's www.novokizol, NOVOCHIZOL.
.com.
.ch, because it is Swiss.
Yes.
I will
try and see if I can add it. Show people. Is that it?
Absolutely, exactly. So if you want to take a look at the pig, we have videos. I mean, this whole thing is very surreal. So if you want to take a look, he's going to be our mascot. He's going to be our hero. Hopefully it has a purpose. So yeah, it's happening. It's happening. Crisis is opportunity. If we can share it with the right people for the greater good, it will be such a gift.
Wonderful. Thank you again so much, Vanya, and I would really hope that we speak to you again soon.
Thank you, Philip. Talk to you later.
Talk to you soon.